right. Welcome to the Human 3 podcast. I'm Sam. I'm here with Brom.
Hello.
And Martin Jacko, the founder and CEO of Aperture Therapeutics.
Hi, everybody.
Welcome. So we're here to talk about Aperture, but first, I just want to tell everyone, Aperture is a company in our portfolio that we invested in last year, and Aperture is focused on developing Antisense Oligonucleotide therapeutics, also known as ASOs, for neurodegenerative diseases like ALS. And they do this by leveraging insights from genetic profiles of naturally disease-resilient individuals. So, can you tell us a little bit about Aperture in a nutshell and what it means to mimic protective gene variants? What is a gene variant? Boil it down for us.
Right. So really, we have started the company understanding that, it's been really hard in the past to develop therapeutics for neurodegenerative diseases. And we took a unique approach where we actually find gene variants. So those are sort of small changes in the genes between the individuals to understand, why different people experience these differently. And we can use this genetic data from the patients to understand, why some people deal with the disease better than others, and then develop therapeutics that actually mimic those protective variants to actually ameliorate disease.
That's great. So basically, one of the reasons why Alzheimer's disease, frontotemporal dementia, ALS, one of the reasons why they're so hard to treat is just because they manifest very differently in individuals. And Aperture is really taking this approach of understanding who's good at, I guess, better dealing with these diseases than others, and trying to apply those biological insights to developing a drug that can be used for everybody.
That's right. It's sort of like democratizing, the protective variants and understanding, the resilience that is really unique to few individuals and making therapeutics that you can generalize the protective effect to everybody.
Like taking the good stuff out of the resilient individuals and putting it in a drug for everyone. That's great. Let's talk a little bit about the first drug that Aperture is going to develop. You're going after ALS. Can you tell us a little bit about what ALS is and how ALS generally manifests differently in different individuals based on their genetics?
Yeah, so, ALS is one of these neurodegenerative diseases that's been sort of the holy grail for a lot of people in the neurodegeneration space. It's one of the most severe types of neurodegeneration where, people are typically expected to survive 2 to 4 years. And so, very severe disease. Currently, no treatments to speak of, especially for the sporadic types of disease that affects most of the patients. And the thing is, if you look, as I mentioned, if you look at the disease, you would expect typical survival 2 to 4 years. But the reality is that the the patients with the disease survive anywhere from 1 to 50 years. And so, again, at Aperture, we're trying to sort of understand, why are some people surviving longer, and then develop treatments for the disease based on that knowledge.
Yeah, and you mentioned this word sporadic, and for those listening who aren't familiar, generally when it comes to neurodegeneration and diseases like ALS, you can bulk them into 2 categories, sporadic versus familial. Can you break that down a little bit?
Yeah, in some ways, that categorization is, slightly arbitrary, where we basically call the types of, or the patients where we don't understand that they have a family history of the disease sporadic. And so in the case of ALS, there will be about 85% of individuals. And about 15%, you basically know what the gene is that is causing the disease. And, you can categorize those further then.
So, interesting. So basically, even though familial ALS, of course, there's the family history, and of course, there's surely a genetic link there in 100% of familial cases, would you say?
Yeah.
But only in 50% of sporadic cases do we actually know if there's like a gene mutation that's driving it.
We actually call the sporadic ALS, we call it sporadic because we actually don't know the origin of the disease, so we wouldn't even know what is the gene causing the disease. In the familial, that is about 15% of the patients. There are hundreds of mutations that we know are causing the disease and are inherited in the population.
I see. Okay. So 0% knowledge on what genes are driving ALS in sporadic cases.
I see.
Yeah, interesting. Sorry, I thought you were going to say something again. I don't know what I'm hearing here.
I didn't say.
When we were talking before, Martin, we were chatting about this idea of neuroinflammation more generally and how this has been an area of interest for drug developers for the past couple of decades now. And when you were talking about your story, you said that neuroinflammation picked us. I'd love to hear more about that. How did neuroinflammation pick you?
Yeah, so in many ways, when we started the company, we took an unbiased approach where we look at what gene variants are associated with protection in the disease. And I actually come originally from a background that is primarily focused on the neurobiology and neuroscience of the or biology of the neurons. But when we look at the top gene variants that are actually affecting the disease, they were all associated with inflammation. Now, to some extent, it's not surprising because, as you mentioned, neuroinflammation was out there as a sort of pathology that is observed in all neurodegeneration, but in the past years and decades was often considered to be sort of a marker of disease, a consequence of degeneration that is, originating in neurons. But there's more and more recognition now that you actually do have targets in the neuroinflammation or neuroinflammatory pathways that drive the disease, and that's what we've seen in genetic data. And really, the genetics data suggests that the difference in the immune system and inflammatory pathways between the individuals can actually affect how you experience neurodegenerative disease and how severe it gets, how long you survive. And so that kind of makes it a prime candidate for us to actually develop therapeutics targeting those pathways.
I think what you said, it's a really important piece of nuance that I want to highlight, because I think in a lot of conversations that I've had with maybe tourists of the neurodegenerative drug development space, it's a very common thing to say, well, of course, neuroinflammation is a byproduct of the disease. But we don't hear a lot of nuanced discourse around how there are targets that actually drive the pathology too. And obviously, I think to kind of step back a bit for listeners who may not be like as technically familiar, but when you have neuroinflammation, that can also be comorbid or coexist, if you will, with other types of like autoimmune diseases as well. And so I think it's really interesting. And I'd love to take time later to chat more about how the Aperture platform is unlocking novel insights over time as they develop drugs. But you mentioned that you started in like neurobiology and the neuroscience of neurons themselves. We'd love to hear about your path to Aperture and kind of what got you here in the first place.
Yeah, so I'm originally from Europe, from Czech Republic, and actually have studied genetics for my master's, for my bachelor's. And one sort of unique thing, I actually come from a town that is considered to be the birthplace of genetics, where Mendel did his original sort of P-crossing experiments, and so I have sort of deep background in in genetics and sort of interest in that field. But I was lucky to actually come to US as a grad student to Columbia University, where I was in a lab that actually have discovered how to make stem cells into motor neurons. And so motor neurons, for those that do not know, are neurons that are sitting in your spinal cord, and they actually extend all the way to your muscles and actually drive all of your movements. And they are the neurons that are primarily affected in diseases like ALS, where you actually are, losing the power of the muscles and are become basically paralyzed. And so, coming outside from that lab, had a pretty good knowledge of motor neuron biology development. I actually work on projects that discovered the molecular drivers of how motor neurons are maturing and aging. So there was pretty good background, sort of understanding the basic biology of these, very specified cell types. Went to do my postdoc at Stanford, where I actually developed a couple of technologies to do high throughput development of therapeutics that led me to biotech. And so, part of the story is me coming to Bay Area. I got very excited by the biotech industry, and had a couple different interactions with the different founders. Actually ended up joining or cutting my postdoc short and joining a company called Serotiny. There was a great experience from both, scientists being in the biotech, but also getting some operational experience. Being one of the first employees there, had to wear different hats, and so I was involved in a bunch of partnering efforts and IP strategy, and that led me to Bridge Biopharma, where I was working on their business development team and was able to build couple different companies in the genetic disease space. And I through that career, I kind of went through a couple different working on a couple different diseases and a couple different therapeutic fields. But I've seen that growing number of genetic data in the patients with ALS and other neurodegenerative diseases, and it really seemed like a great opportunity to actually apply some of the knowledge of the genetics to actually find new targets And, that's sort of how Aperture came about. I was, throughout my career, actually stayed in touch with a lot of my mentors, throughout. And so, kind of put the heads together with a couple leaders in the field and, jumped into Aperture to actually, finally solve diseases like ALS.
That's awesome. Yeah.
Yeah, I mean, in terms of solving diseases like ALS, what is the current state of the art in terms of treatment for ALS? Like if I get diagnosed with ALS today, is there anything that can really be done, or is it we pretty much just watch and wait for it to progress?
It's quite sad. There have been more than 150 drugs ever tried for ALS, and most of them haven't done much for people. Now, if you're one of those, it's hard to say lucky ones, but if you have familial disease, familial ALS that has one specific mutation in a gene called SOD1. There is now a company called Ionis that actually develops an antisense oligonucleotide that can actually target those mutations and potentially extend the life. But that's really only the case for 1% of the people with ALS. It's really rare. The rest of the people, that would include the rest of the familial and the rest of the sporadic, really there is just one therapeutic or two therapeutics that were ever approved, one of them called Riozol, the other one Edaravone. The first one that is sort of most used by most people, Riozol, was approved in 1995.
Wow.
And, based on that trial, it extends the end-stage survival in the end stage of disease by 2 to 3 months. And so, really the idea is most of these people take it because it's generic, is relatively cheap. It doesn't have a severe side effects, but you wouldn't feel the difference. Right. And so really the benchmark for helping people with ALS is unfortunately quite low, but that also sort of stirs up, a lot of effort to actually make an impact. And, that's where sort of Aperture enters. We're trying to really, get in and develop therapeutics where we would, significantly improve life of people with ALS.
Sense. It's amazing that over 150 drugs have been
Crazy.
trialed. And
I think,
none of them have done anything super substantial.
Sometimes that actually, this is where you get the conversation with investors, that can turn some people off, and it might seem like a graveyard of effort and therapeutics. One of the, lucky things, and I actually had time to look into all these therapeutics and actually before starting Aperture, actually went over every single drug and every single trial that was ever done.
It's a lot of data, right, that you can use to inform your own approach.
Well, the crazy thing is what people have tried. I wouldn't discourage everybody. There are definitely some therapeutics and efforts that have seemed really promising. But there are some efforts that were really shots in the dark, and I think that turned a lot of investors off. There were drugs for HIV, there were drugs tried for ALS, vitamin C, vitamin D, fecal transplants, and all of that 150 drugs would actually be filled, with that category of, let's try.
Like random, let's give this a shot. Interesting. And were any of those 150 experiments similar to what you're doing, where you're being informed by the genetic profiles of people that seem to naturally be able to live a lot longer with ALS? Or is this a totally brand new approach?
To some extent, it's a totally new approach. It's not we're not the first ones to have a genetic approach to ALS. There are companies, there were leading efforts like, that aforementioned Ionis that have developed and had success developing actually antisense oligonucleotides for SOD1. That actually goes after those specific subsets of patients with specific mutations. But, starting a company in that field is quite challenging because you're looking at very small market that you can address. There are other companies that have tried to focus on disease risks, and those are sort of genes, sort of the opposite of what we're trying to do. And happy to talk more about those if you want. But we are kind of trying to carve out a space in those protective genetic variants.
I think it's worth talking a little bit about why these 150 attempts, although some of them it sounds like were just kind of shots in the dark, like you said, but why some of the more concerted efforts haven't been successful. And in my opinion, and I'd love to hear if you agree with me or not, and maybe if there are other things that you think contribute to the reason, but in my opinion, I think it's a number of things. I think that there's a lot of work that can and should be done at the discovery stage that just isn't. I think it's the quickest to clinic, whatever we can get into the clinic, the fastest, the easiest, and I guess the cheapest way is what we should do. And I think often those attempts haven't segmented population based on genetics for sure, but also what they're doing in the end when the trial fails, it's hard to even like analyze the data in a meaningful way if they didn't do any genetic profiling of the patients. And so I suspect that that's part of the reason. I also think the modality is part of the reason. I don't necessarily know what the majority of these trials were in. I'm assuming a lot of them were small molecules at first, maybe some antibodies. I'm not sure. But if you had to, state why you think they've failed so miserably, obviously, I think we can agree that your approach is going to be more informed, but are there any other main reasons that stand out to you?
Yeah, I think, you summarized and picked up on a lot of those reasons. And I think, things like stratifying population and ALS and Alzheimer's and Parkinson's and other age-related neurodegeneration are really a group of diseases rather than one disease. And so that definitely is sort of a factor that plays into it. Another factor is, we weren't really sure, or the field is sort of maturing in how do we measure success. And, a lot of the biomarkers and other sort of measures of like, is my drug working, are only available these days. I think what we focus on, and I think what is one of the major reasons is, even before going into clinical trials, the way we measure whether, or the way we predict whether this drug will work is largely depending on animal models or cell models, right? And people are trying to sort of continuously evolve and develop better models as different sort of more complex cell culture models, iPSC derived models, and there have been a couple different mouse models that have been developed as well, but they're turning out, as you can tell, right, from no real therapeutics being approved. They're turning out to be not great predictors of therapeutic success. And so a lot of times, the way our industry works is you would actually take new chemical or chemicals that have been established, do a bunch of studies and try to see positive results. And that will be sort of a validation for you to get a large amount of capital to go into clinic. And that sort of path haven't proven to be working. That doesn't mean that it's not working for other diseases. It really seems to be something more specific to age-related diseases and neuro. That this model just is not operational. And so again, this is where we really started the company. I am biased towards genetics because that is my background and towards neuroscience. But, we have started the company really asking sort of from first principles, what can you do and what kind of data can you gather to actually treat this disease? We went more than approach first. We went to problem first, right? How can we actually solve ALS? And that kind of boiled down to, really genetics is one thing that you can, that you can trust to some extent. A lot of these disease models are sort of secondary confirmations. And so I think, models is one big reason for a lot of failures and poor prediction of what works in humans.
That makes sense. When it comes to looking at the genetic factors that seem to predict if someone is going to live a lot longer than someone else, like Stephen Hawking, for example, it seems like he lived a very long time. Probably is it
close to 50 years?
Yeah.
Yeah.
So is it, based on what is it a small set of genes that are somewhat able to be identified, or is it a lot of factors that kind of line up that determine whether or not you live long once you get diagnosed?
Yeah. Well, I think it might end up being a lot of factors. Just to be clear, the way we actually look at the data is not too deep. What we're basically looking at, we are bulking all the patients and trying to figure out what are the common disease modifiers.
Right.
And I think based on the origin of the disease and, Stephen Hawking, I'm actually not sure exactly why or whether there is any explanation for, did he have genetic ALS or was there any environmental factor? I think that there might be specific reasons for individuals that might be sort of more protective than others. But we're sort of taking the broad approach where we actually just bulk everybody. And then to some extent, it really floats out the disease modifiers that have the biggest effect.
Sure.
Right? Because we're looking at everybody at the same time. And so then our therapeutics will be applicable to everybody and, we'll provide some treatment effect for everybody. Yeah. But in the end might be, hundreds of factors. What we see is more like dozens.
Gotcha. So there, even though there may be hundreds, there's still a small subset that seem to drive the most of the variance in outcomes.
Yep.
Okay.
There are so many rabbit holes I can go down, and I'm trying to figure out what the next one should be. I think I'm not sure if you've seen this ALS influencer online. Her name is Brooke EB. Have you seen her before?
I haven't.
So she is a woman who is in her mid-30s who was unfortunately diagnosed with ALS, I think in her early 30s. She works at Salesforce and she has this huge following. She's a really strong advocate for ALS. And I remember on one of the adcoms for a recent drug that was trying to move through the new drug approval process with the FDA for ALS. She was there, like, I guess, testifying as like an expert. And I forget what drug it was, but there was a lot of unrest and disappointment in the ALS patient community because this drug wasn't approved. Do you recall any of these, like, I guess, debates at adcoms, or like, why do you think that comes about?
Yeah, it's honestly, ALS is a very difficult disease, and actually a lot of these patient organizations, a lot of the influencers actually have a really positive influence on the advancement of therapeutics in ALS. It's quite crazy if you actually look. There are other diseases that are quite similar, like FTD, frontotemporal dementia, much less sort of public support for reasons I don't completely understand, but, it really kind of gives you an idea of like how much can individuals outside of the field do for a specific field and for disease.
It would be hard to be a FTD influencer, because your brain starts kind of.
Well, yeah, that.
Well,
but
you can be a family of.
family.
That's true.
And so overall, the patient organization super critical. For us, we are focusing on ALS partly also because of the availability of the data that is provided by the nonprofits and the different organizations. This is not the government. I mean, government is behind, some of this funding as well, but it's really not a pharma or industry effort that allow this. It's really, all the patient organization. That said, there have been some controversies there before where, drugs got approved because of pressures and they didn't end up panning out. And it really seems, from my point of view, being an outsider, even before starting Aperture, that the government, the FDA, has, in the previous administrations, I'm not commenting on this particular one, have done actually a really good job and have sort of put the best faith, decisions forward. And, there was really like open discussion back and forth. And, I think the process works for the most part, right?
Yeah.
But it's really, I don't think we're hindered by the I don't think the reason that we don't have therapeutics is the regulation. The hint is the science.
It's just that the drugs are hard to make. Yeah. I agree. And it is frustrating because you hear a lot of people that, have negative, less than stellar opinions about the FDA. They'll blame this stuff on regulations. But the reality is like, even if there were no regulations, you still wouldn't be able to make a drug that actually did anything.
I think the problem with the regulations, with the FDA is, when you have a therapeutic that is, you need to screen your eyes and, try to figure out whether there is an effect or not. You have disagreements.
Of course.
Right? So if there was a drug that was really clearly working, it would just fly through.
Right.
So.
Right.
I think in this example specifically, it was clear that it worked for some individuals and not others. And this goes back to what you and I were talking about. Like, if we don't have clearly defined parameters of subpopulations, like maybe in the future the FDA comes up with a way to try and subsegment trials and like look at new biomarkers and then run a second set afterwards. I feel like that is probably why there was a little bit of unrest in the patient community. But that being said, too, these biomarkers that are used as clinical endpoints to measure the efficacy of the therapeutic, I feel like maybe those ranges are different for different individuals. And I think that complicates things even more. Yeah. You said something that was really interesting to me about how you're not going after a pathway specifically, you're just letting the data speak.
Right.
And I think it's worth noting for the audience that in central nervous system disorders, in the drug development world in pharma, very often we have these cyclical trends where companies are willing to, like big pharma companies are willing to look at smaller companies who are going after a specific target, let's say. And I think that unfortunately negatively reinforces this dynamic where doing new drug discovery with truly novel targets that could be disease modifying is not encouraged. And so it seems to me like one of the reasons why I was so excited about Aperture when I met you is that you're really trying to flip that on its head. And you're actually trying to do the work at the beginning and quickly show that this can be modulating disease in a way that hasn't been before. How do you feel about just the future of neuroscience? You know, we've seen the mass exodus of neuroscience 20 years ago from the pharma world. Now we're seeing a huge resurgence of interest back into neuroscience. What is your take more generally on this space, and what are you hoping for?
No, I think that can be partly answered by, some of the new technologies and modalities that are now available. Again, this kind of comes back to how we are used to think about science and therapeutics development. Pathway meaning, specific molecular, interactions, and effects that those have on the cells. We always are used to, within a specific pathway that is responsible for a certain function in a cell, trying to find a protein or other molecule that we can actually develop a drug for. And so that is how industry has worked for a really long time. And once you can actually find a molecule that you can drug, there is a lot of focus from a couple different companies and all working on, a different mousetraps for that same molecule. There are new technologies now, like these antisense oligonucleotide therapeutics, that are basically short pieces of RNA that can actually modulate function of specific genes that actually don't rely on the protein biology. They are relying on the RNA biology that is, quite universal between different genes. And so what that really allows you is to avoid a lot of the pitfalls of the complex pathways, and it allows you to pick whatever you want in the pathway now. You don't necessarily need to focus on, specific proteins that you can typically develop a drug for. And so that was kind of one of the reasons actually for starting Aperture. We actually look at the genetic data even before we started the company, and then we look at what the targets are, and nobody was ever able to drug them really well. And so, the idea of starting the company was, using these new modalities, we can actually develop now drugs that hit these targets on the RNA level. And so, I see that combining genetics and new modalities, mRNA, short RNA like oligonucleotides, that kind of completely opens up a new space where, you can pick whatever target you want pretty much, and you can lean on the genetics to tell you where to go and where in the pathway is the best, is the best target to intervene, and then develop the therapeutic around it rather than being sort of limited by, what your chemical abilities are.
Yeah, I think, when you were talking about your background, you mentioned that you were like doing different things in age-related disease, and then you did a postdoc looking at high-throughput screening. I think your background has lent itself well to what you've built in the platform at Aperture. I mean, again, for those who are listening that don't know what high-throughput screening is, it's the ability to use different technology to really quickly understand whether or not a specific drug can be useful in a disease context. So it often takes a lot of time and money. High-throughput technologies are a way in which you can accelerate that. But when it comes to getting more specific about the models, which is, I believe, what the main thing that you're trying to do is look at these human cells for these insights about resiliency. Trying to adapt that to a platform and make it high throughput is no easy feat. And so I think in a nutshell, it seems like you're trying to iron out the kinks of a platform that would be the beginning of like this, I guess, grand flywheel of learning about the neurodegenerative biology in a drug discovery context. And that's basically what you've been working on now. And I don't think a lot of people realize that when you inform your decision-making process very early on in the drug discovery process, it makes such a difference in the end product. Like it even will inform which mouse models you use, even if they're not the greatest models. Sometimes the decisions that come before that actually make the outcomes better. How do you think about that in a very vague sense? Like, would you agree with that, or would you say something else? Like, do you think it's correct to kind of characterize your platform in that way?
I think so, and it's sort of difficult situation sometimes to be in because, you are basing everything on the genetics, and to some extent, you don't actually need, we have mentioned early on that we don't necessarily believe the animal models, but
Right.
you need to prove out your drugs, your drugs work. And so, this is always sort of push and pull situation where, you are trying to get the validation in those animal models without relying on them. And so, I think, the platform is really based, anchored in genetics, and we're basically trying to do a minimum, as little validation as we have to actually have the additional confidence in the high throughput cell-based screenings, and then validating things in the animal models. But I honestly see that in the future, once we get comfortable with designing therapeutics and developing drugs based on data and prediction based on human genetics and AI machine learning that can make significant predictions about how certain drug will perform, we would just basically use the mouse and the cell models, less and less, and we're totally cool with that. We're not very excited, injecting a lot of mice with drugs to test whether they're working in humans. And so, that's where I see the future of the field also going, is sort of leaning more and more on the data.
Yeah, no, totally. I guess zooming out from the technical a little bit, we were also talking before about some interesting things that are happening in the broader ecosystem right now. Specifically, there are a few drugs that once they read out, we might gain some insights. We could talk about that, but I think we were also talking about just the availability of genetic data more generally. And where do you see genetic data going in terms of everyone's therapeutic pipeline? Like, I feel like most individuals who are at the cutting edge of drug discovery right now know that they should be using some sort of genetic data, but not everyone is. So
Yeah.
do you think that some of these consumer-facing companies, like, I don't know, 23andMe, for example, do you think that there is a real chance at leveraging that data to create powerful therapeutics, or do you think there is a different type of data set that needs to be collected?
No, I think so. For us, again, we were quite lucky and also very purposeful about working on a specific problem because of the availability of the data. And that was, again, because of the activity of the nonprofits and government support and sort of other sort of interests in the disease. But you can see that there are quite a few more diseases that are affecting the broader population. These are not necessarily rare diseases. Even things like Alzheimer's, where the genetics would actually be super useful to actually develop new drugs. And the more and more people are sort of comfortable doing genetic profiling and potentially sharing it with people that are developing drugs, I think the better therapeutics we will have. We were a little jealous that 23andMe might potentially go to some of the data might potentially go to Regeneron. I think that is definitely a great asset to have. But we see new companies in the genomic space that are emerging that provide maybe potentially even better data. In the case of 23andMe, they actually did what we call genotyping for specific variants, where they focus on very small portions of genome that we can interpret, and that's sort of a prudent commercial strategy because you're not sort of wasting money on the entire genome of the human. But in the future, we'll be able to interpret more and more on the genome, and so I think companies that do entire genomic profiling will definitely have better data than even 23andMe has. And so we're definitely looking at some of those efforts and watching it very closely, and might potentially make some deals around in the field to actually be able to expand into other diseases as well.
Yeah, I was going to ask, so is that why you think that 23andMe more or less failed to get any drugs developed? Because if you go back even a decade and look at 23andMe's media, they talk about developing drugs. That's kind of like one of the things that was always perpetually on the roadmap, but I don't think they really made any progress at all. Is it because they didn't pay attention to the entire genome sequence and they only focused on those small bits that you think they were unable to be successful in that area?
Just to be transparent, I don't know why they
Speculation.
why they failed to develop therapeutics that would be successful. I mean, a lot of times, and this is getting this is a tangent, but sometimes things make sense and it's pretty straightforward to develop. It might seem like it's pretty logical to do certain things and develop therapeutics. In the end, it's a human effort and it's hard, and it might fail for other reasons than the data.
Right.
There is also, they certainly had the samples to actually get the data that they needed, is my answer. And, and build up, and they certainly had the capital to actually develop therapeutics. So I'm not necessarily sure why.
Maybe it was more business and human reasons than scientific reasons.
That would be my guess, but I'm honestly not sure.
So how do you get the data for your experiments now? Is it through patient advocacy organizations or
right? And Regeneron is willing to pay for
so
the
they
data.
must think
So I don't
worth
think it's all
something.
bad.
Yeah.
I really think it's probably quite valuable data. But we are super super grateful that we're able to work with nonprofits. They're just overall like open to sharing this data and under specific agreements. I think another thing that is super critical, and obviously companies like Regeneron are very aware of that, is a lot of the genetic based approaches previously were based just on genetic data. And so, people were sort of trying to figure out, what is the difference between a patient and a healthy individual, and what are the gene variants that you can find in the patient that, distinguish them. If you want to take our approach, the critical part of data that is missing is the clinical profile and sort of, biomarker profile or patient history or, individual history. And so I think that that's where the value will be, once people will be able to combine their medical records with the genetic data. That is the most valuable data to be having. Yeah.
Makes sense. Okay, cool.
Yeah, I think there's a lot of discourse in terms of trying to, I guess, integrate EHRs, other types of behavioral data, also other types of data that can be collected from like consumer tech that's like a wearable, for example. And it's all fine and good, but when I think about these like thoughts around the future of precision neuromedicine, it's always difficult to figure out like who is going to be the one that will actually take that step and bring all of that together collectively. That isn't a for-profit, because I find that once you start to add multiple modes of data, the R&D work becomes so great that it almost seems difficult at a certain point to kind of integrate all of it. You have to do it one at a time, and if you're a company, you can kind of build towards that. But I still feel like it needs to be some kind of central organization that collates all of it somewhere, maybe like a pre-competitive consortium of some sort. Yeah. Have you read this review paper? Harold Hempel, the CMO of Eisai, was the first author on this paper in Cell from 2023. I think it was The Foundation and Architecture of Precision Neurology and Psychiatry. Yeah. Okay, I read that and I was like, wow, we have all the tools. Yes. He clearly maps them all out there. But we haven't been, as a society, organized in such a way where we're incentivized to collectively leverage all of that data. Yeah. What is the, I guess, future in your mind? Like, do you think it's possible for a company to eventually do this in the next 10 to 15 years, start working with other types of, data collection companies, and then eventually integrate all these tools together? Or is it going to take like a collective pre-competitive effort to kind of get to a certain point before we can actually do that? Because data ownership, I think, becomes also the next question too.
I mean, it's a great point. I honestly think you're completely right. A lot of the questions that we're trying to solve and a lot of the therapies that we're trying to develop, again, the problem sometimes not the science. It's more of a societal problem, or it's more business problem or a structural problem. And, again, so having access to the data, I think for some of these severe diseases, the nonprofits and other organizations will for sure play the most important role in actually collating all the data. And, that is sort of their role, and that is what they're doing. But, then there are diseases where, not everybody sort of might be, sufficiently incentivized to actually share their data. And, those might be diseases that are maybe not that severe, and that, you would actually benefit from having the data, but basically people are not incentivized and not so invested into sharing their data. And, I'm not sure if, you would say that you see the government, being able to actually create, a database or anything like that, or even that's something that we want and it's desirable for us to be having, but maybe in the future, if genomic profiling and the cost of sequencing will become something that is not prohibitive, it will be something that actually individuals have and own, and it's something that is associated with their clinical record. It's part of their clinical record, and they can they can basically opt in or opt out from some of these studies, and you don't necessarily need to have snapshots of. You don't necessarily need to have genetic data from everybody. Snapshots of data might be very useful for us as well, and so I think that that's where it might be going, where genomic profiling for individuals will be something that everybody does.
That's really interesting because, I'm thinking about this. Of course, I don't know if the government would be the organizer of this hypothetical
Hopefully
thing
not.
that I'm talking about. I think it's more like this. I would imagine, if anything, a couple of pharma companies that have a collective interest in a specific disease area are willing to come together in a pre-competitive consortium. But what you're saying is actually a lot more interesting to me, because even with myself, I see that I'm able to collect more personal data and monitor it. I even have a spreadsheet with all of my personal health data.
And partly it's such a kind of neck problem. You see a lot with genetic diseases where, you want the data to develop the therapy, but when you develop the therapy, a lot of people are interested in getting their data to see whether the therapy makes sense for them. And so you can imagine that, your genomic profile would actually be very helpful for your physician to actually tell you, hey, this and that is available to you, or we should watch this and that biomarker because you're at the risk at a certain disease. And so I think it's sometimes an imperfect sort of dance where we're trying to have the data to be able to develop the diagnostics and to be able to develop the therapeutics. But I think as we kind of move in a stepwise fashion, people will actually have an incentive to get their genomic data, because that means services for them, right? It's not just they're not just doing it to help the pharma companies, they're doing it to help themselves. And I think that kind of aligns the incentives over time, where you are now you actually want to get your genetic data and share them.
So in practice, does this look like all of us in the future having our genomes, and then as we age, maybe going to a specialist who can diagnose precisely what's going on, and then maybe if for whatever unfortunate reason I need to come to Aperture in the future, you can perhaps design like a combination product just for me? Or,
ideally we actually have it off the shelf, but we can sort of create a cocktail and mix for you, and we monitor you because we understand that the way as you age, you are at risk at some medications, and then hopefully we get to a point where a lot of it is maybe not completely preventative treatment, but we can actually intervene early enough so that you don't experience the disease in a way that actually disrupts your life. So
yeah. Do you have any thoughts? I'm just going to keep going if that's okay. Yeah, it's interesting. I think about this a lot, and over time, obviously we would like a world where we have more personalized medicine and we can intervene earlier. But going back to some of the specific challenges, a lot of this is hinged on models, biomarkers, et cetera. Do you think in the future, say 5-10 years from now, Aperture is generating leads for experimental biomarkers for disease that eventually become new endpoints in trials?
Well, it's funny you say that, because we actually do that now.
Oh, interesting.
And so, a lot of the targets that we go after is sort of an uncharted biology, and we don't necessarily want to rely on, biomarkers that have been there. And honestly, in diseases like ALS, there is one. Right. And so in many ways, we are actually making effort and sort of putting capital behind actually develop some of these new biomarkers that will actually allow us to monitor the activity of the targets and the drugs that we're developing. And, I think that's also like super active sort of part of science and what we're developing, trying to figure out, what we're actually affecting in the body. Because, again, we go after targets that nobody has ever, went after before. And so, you kind of are sort of forced into that also to some extent.
Yeah, that's great. I mean, it sounds like to me you're trying to hit on all of the major challenges that create a failure mode for neurodegenerative disease companies usually, and preemptively like trying to find solutions for it before.
As much as we were sort of very excited company and try to do ambitious things, we also some of the biggest skeptics, and a lot of us actually have been in the field for a while, in academia or just working on the business side, and we see that this is not an easy field, and so we're very sort of critical about ourselves and kind of trying to plug all the holes as we go forward. And part of that is leaning on genetics, part of that is is actually looking for these biomarkers.
Informed optimism is the best kind. You don't want blind optimism. And
Yeah, that's right.
It sounds like you guys are more on the informed side. We have plenty of time.
I was going to say, in psychiatry, this concept of developing transdiagnostic biomarkers is a big thing. So basically, can we develop a biomarker that can be used as a diagnostic criteria, not just for example, depression, but also generalized anxiety? Do you think that based on the clinical or the experimental biomarkers that you're looking at co-developing with your therapeutics, do you think that some of these will be transdiagnostic? Because there is this common unifying pathology between certain types of ALS, FTD, and PD, as you've mentioned before. How do you see the biomarker development informing future indication selection, so to speak? Do you think that there is something there?
Yeah, 100%. So we actually, when we develop these biomarkers for clinic, we actually don't necessarily wait all the way to clinic. We actually track them during the development of our therapeutics, and so that we have a good understanding, we are not picking a pathway. And so in some extent, we're trying to actually understand what pathway we're hitting because genetics pointed us to it. And so looking at these biomarkers oftentimes helps us to understand, this is what we're actually doing in the cells. This is what the genetics pointed us to. And then we do look at, other diseases. And actually, recent example was, we actually realized a bunch of the biomarkers that we look at are things that are coming up in traumatic brain injury, as well as a post-stroke dementia. And so, they suddenly are sort of being, quite interesting indication to go after because we see that, those biomarkers are popping up. And so 100% we're looking at biomarkers that are sort of, appearing in a couple of different diseases at the same time. And that helps us to, from a business perspective, makes great sense because you're now able to apply your therapeutic to a couple of different indications. But also from the science perspective, you're sort of very targeted.
Yeah, I mean, the brain is just so complicated. Obviously, we've talked about how all of these disease categories, like Alzheimer's disease, is really just like a collection of subtypes of different diseases that we don't yet have the tools or the language to describe. And so if we think about it, potentially hundreds of different types of subtypes of disease. But then again, there's also a lot of common biology. And I think it just goes to show that trying to pick a pathway to start with, historically, that's probably one of the reasons why it's failed, because this pathway that, for example, in the past, people have gone after tauopathies or alpha-synucleinopathies. And I think that that's fine. But when you're going after something that is so common and shared amongst a collection of a wide variety of disease, it's hard to even fathom how you could select a clinical population that would respond. At all.
I think also, the understanding of pathways in abstract, really the biology, if you imagine, is or the cell, if you imagine it, is sort of a huge network. And when you talk about pathways, just, a branch in that huge network that's interconnected with everything else. And a lot of times we sort of abstract it away from the rest of the cell, but there are a lot of relationships that we don't fully understand. And biology is just freaking complicated. And so, a lot of times there are different feedback loops and, there is a lot of complex relationships that we don't completely understand. And so, pathway in many ways is very reductionist approach to looking at the problem. And so I think that's, pathway might mean 100 different targets. I think that's why, a lot of times you're sort of guessing really rather than, being very targeted on a specific drug target.
Yeah, totally. I have a question for you, and this is more me trying to learn a little bit more about the nuanced biology here. But sometimes I think about, in biology, we have a cascade of things that happen all the way from the translation of mRNA transcripts down to the final pathology manifesting in the disease state. And so for those listening, there are many different proteins that are kind of identified along the way, or molecules that people try to go after. Mm-hmm. When you think about groups that are going after very downstream proteins to treat disease, is it a fair assumption to think that at best they're just treating symptoms? There's obviously not going to be any kind of regenerative situation, I think, in those contexts. And I try to think about it like an equilibrium. If you focus here, you're probably going to push them to a state where they're at least not progressing as much. But if you push here on the upstream side of things, you're more likely to actually get some kind of true disease modification. Or is it more complicated than that?
I think I actually do pretty frequently use sort of the terms upstream and downstream, but really, again, that's sort of a relatively arbitrary, nomenclature. And a lot of times you realize that the downstream is actually regulating the upstream, and then it's a loop. And it's really hard to say. And so that's why, in a way, we also are playing ignorant, and the way we approach the problem is to say I don't know. I don't know how this works, but I do actually have a measurable way that actually points me to things that make a difference. And so we're not necessarily trying to explain or even try to pretend that we understand all the different pathways, but what we do understand is hitting a specific target results in specific biomarkers to be regulated and a specific phenotype in a human. And to some extent, it's for us that's good enough. You see it with many companies, with GLP-1s, with PCSK9, and a lot of beta amyloid. Beta amyloid is not a great example, but a lot of times you end up with the drug that you don't fully understand how it works. But if it works, you just go with it.
That's interesting.
Sorry,
or
choking on my coffee.
So I guess a simple way of trying to think about this is what I just described was our best guess at a map of what's happening. But what you're saying is that we should probably just go with what we can measure and perhaps use the map as a guide. But in essence, 100%. We're kind of building a new atlas instead.
That's right.
Okay.
Yeah.
That makes a lot of sense. Cool. Thank you for that. In terms of, what you're up to for the next little while, what are you the most excited about? Are there any kind of market catalysts that you think will change people's opinion on neuro for the better or for the worse? And obviously, I think that people listening should not generalize if they see one bad outcome in the market or a good outcome that, everything in that category is going to be good. But generally, neuro has been this area that's quite sensitive. Either investors are like fully bought in or they're not. And so I'm curious to know if there is anything that you're looking forward to, both in terms of what you're working on, but also in the market as well.
Yeah. I think, once you get a positive readout and an approval in the disease modifier space, and we actually sort of see it now, maybe not necessarily with investors, but with pharma, there will be a huge amount of interest in the next drug that actually, adds on top of that effect. And, basically you validate the approach. And now, it's sort of like, in oncology, if you develop a chemotherapy, then you do, and that works, you want to develop the next one, and next one, the next one. Similar with GLP-1s, you develop the first one, then you kind of come up with the consequent or the subsequent generations. And I think once we actually, get an approval for a disease modifier in ALS or any of these neurodegenerative diseases that we're looking at, it would just spark a huge amount of interest because suddenly these are treatable diseases. And, there are some caveats in whether or not, there are therapeutics that, have treatment effects, but for the most part, there hasn't been much. And so, we actually see from pharma where there's already assumption that some of these will work, and there's interest in like, okay, what is the second one? What is the third one? And, we're lucky with that approach that we're taking that is pretty economically efficient generating these oligonucleotides. And so we're building a whole pipeline of targets. And so, we'll be sort of ready out there with, pushing our first one into clinic. And you ask, what we're excited about. We're excited to actually push our first drug to clinic within about 24 months, and that would be sort of a very exciting time to actually see, how this, approach proves out, and then, we're there ready with the second generation, third generation drugs as well, and hope that, in 5 years from now, you'll have multiple therapeutics that will actually give you, extension of survival and slowing disease in things like ALS, FTD, Alzheimer's.
That's awesome. We're really excited about it, and we're definitely looking forward to the day in which, there's a selection of therapeutics for people that can be matched to their genetic profile as well. I think that's pretty cool. Did you have any thoughts, any questions?
Well, I wanted to ask what your thoughts on being a founder are, because that's very different from, your life prior, both in the academic world and actually working in the biotech industry as an employee. Like, how has that transition been? Do you enjoy it?
Well,
What have you learned?
In some ways, it's not that different because I was always, ready to dial it up to 10. And so the work is different. I don't think the effort necessarily is different. I was always kind of 100% into what I was doing. And so it's not like I was taking a break before and I'm not taking a break now. I think the difference is, I think the sense of responsibility and sort of being, I was always the guy in the back room that dealt with the operations, with the problems on the technical side. And now I'm the guy in the forefront dealing with, any incoming, and I'm dealing with the problems that other people cannot solve. And so, so that kind of, it doesn't necessarily increase the amount of effort I'm putting into things, but it definitely increases the difficulty. And I think, one major thing that I sort of highly recommend for people and just generally sort of for other founders being excited about, a certain idea and maybe coming out of PhD or postdoc and sort of not having experience in biotech. What really helped me out a lot was having sort of a leadership on a technical side and an operational experience. I ran a couple of businesses before, went to a company as a first employee, so I dealt with a lot of different things, then being on the BD side as well, being involved with, strategy, IP, and sort of different aspects of the business. That gave me like a head start over everybody, and we were sort of able to hit the ground running. And really what I'm focusing on now is basically replacing myself in every area with the person that is better than me. And so we were lucky, getting a bunch of great people in the company. And so I'm kind of, removing myself with few things. And but the problems still come to me. So yeah, that's been sort of a learning curve for sure.
Fair enough. And what about fundraising?
Fundraising.
What advice do you have for other founders that are about to embark on their first fundraising expedition?
Yeah, I think I have, kind of still having some business and a startup background, but coming mostly from the, science. I had sort of the idea of like the data will speak for itself.
Right.
And you will like work hard. And, I was pretty confident about all the technical aspects and being able to execute because of all the experience. And so I was like, I'll just go out there, raise a little bit of money, hit all the milestones earlier than I told the investors, and then I'll go out there and get bunch more money. And it doesn't work like that. And so I have underestimated like how much sort of social aspect there is to building even the biotech company. That I thought is something that is, mostly reserved for, other businesses outside of science, consumer startups, and because you don't necessarily have data. Now I realize they actually have more data than we, right? Because we're trying to prove out our product, 5 to 7 years from now, whereas the consumer people actually are getting feedback from customers, and so. I think building a company and fundraising at the same time is kind of like almost two jobs that you're trying to do at the same time. And so, I think, one of the things you got to be comfortable with is being, told no. And, but also don't get overwhelmed by it. And just, I think that, again, the operational experience actually has helped me because I was very comfortable in, managing the company and the people and the HR and all the problems and kind of, do more learning and growing on the fundraising side. And so, I mean, get advice, get connected, and get ready to get uncomfortable. That's for sure.
That's good advice. And I think just to reiterate what you said, because I think it's a very important point that actually Sam and I have been talking about lately. You were saying that you thought that the marketing or the social aspect was reserved more for consumer companies. And in the startup world, there's all sorts of like slogans and, little cliches about how distribution is more important than product and all that, right? Like that's in 0 to 1 and probably a million other startup books. And a lot of people read that and they think it is only for consumer companies, but every single company has problems with distribution, and that includes biotech companies, that includes deep tech companies. And at this stage, the distribution is how do you distribute your idea to investors to give you more money, right? Yeah. And if you ignore the reality that this is just another marketing thing, then you're going to have a hard time fundraising.
But there is the other side of the coin for it also, and that I have actually sort of struggled with, where for the identity of our company, I was trying to figure out whether there is a sort of different side of the company that we should create to actually present it in a way that is appealing. And in the end, we have decided not to do it and be sort of true to ourselves. And it's more about, really staying true to your story and true to what you're doing and actually presenting the company the way it actually is. Sure. But polishing how you do that rather than, creating a whole marketing campaign that is actually, in some sense fake.
No,
right?
no.
And
you don't want to share anything that's fake, but you still have to tell the story.
Well, fake in a way, fake in a way that you basically do, a lot of times what happens is that you're sort of more grandiose. Not fake is, incorrect or not true, but fake in a way where you basically are trying to gaslight people in like, hey, I'm going to like treat all diseases within the next 10 years, right? And be kind of more, down to the ground and be true to, and then, I think that you actually, that will actually self-select the investors. Sure. Right. And so we actually sometimes, could have gotten investors that, I failed to get in just because the way I presented and pitched. But I'm very happy with the investors that we have because, those are the investors that actually responded to how we pitched the company as well. And so in the end, I'm very happy with how things ended up and, the sort of community we built because we were just true to ourselves. And so, we don't need to pretend anything.
I think that's also a good point that people can take away. It can be very frustrating if you're like in the VC startup fundraising hustle and you see all these people around you raising money off of these grandiose claims that you, because you have technical expertise in whatever domain they're raising for, that those claims are not going to
it's ridiculous
out, right?
sometimes.
And so you can sit there and be like, man, am I like an idiot? Should I
Right.
start telling the same bullshit story so that I can raise money? But I think in the long run, it hurts those people. Because
the answer is no,
because
don't do that.
eventually,
Yeah.
It can.
Because it's great that they raise money now, but eventually people are going to realize that whatever they promise is not happening. And then those investors are going to kind of bail. And those investors are also not sophisticated. They're not going to be able to help. So I think it's better to be more realistic.
I think in the worst case scenarios, you actually see that some of the grandiose will actually seep into the culture of the company and what the company does, and that just kills everybody.
Sure.
Right. And so maybe actually I'm jealous for some people that are able to raise, these shiny, the great, get the huge amount of money based on these like shiny polished tags and pitch it in a way that I think is quite unrealistic and maybe, just ridiculous. But you have to be super careful that doesn't become your company.
Yeah.
It's true. And I mean, we're starting to see a lot of that in bio these days, especially with all the excitement around AI, right? Like you're seeing a bunch of traditional tech investors who have never done any sort of drug development program before put a ton of money into some company who the first slide of their pitch deck says, thanks to AI, biology is now programmable. And we can just develop a drug for whatever we want. And
You had like the little devil on your shoulder the whole time. And like, we have these discussions all the time. Like, should we like be doing a little bit of AI? I'm like, are we actually doing AI? No, we're not putting in a deck.
Yeah.
Yeah, but you are using machine learning, but it's not in your deck. And that to us, like for us, anyone that's doing truly like impactful cutting edge work is using some kind of computational tool and has been. But when it's the main driver of the conversation, I can't tell you how many decks we've seen where it's like AI enabled drug discovery is the first thing, but that shouldn't be what they lead with, Yeah, it's definitely interesting. I was going to ask you, I guess, a little bit about, Brom was asking you about how fundraising has been. What would you tell other founders? What you said in response to that about it's about how you communicate and not necessarily what you communicate. Have you found yourself more in this like translator role where you're dealing with people at different funds that maybe are like technically trained, but they're generalists, and so you have to kind of translate to that archetype and then also translate to the generalist who doesn't have a tech background?
You definitely need to sort of empathy where the people are coming from and kind of translate to different people differently. And sometimes, if you're lucky, the investors are patient and they actually give you more background themselves and sort of where they're coming from, what's important to them, and we're better about asking actually even without them telling us to make sure that we don't miss the target. But yeah, I mean that's what you have to do. You have to be sort of a person that can read other people and trying to understand where they're coming from, what they're interested in, and kind of again pitch the story in a true way, and kind of try to explain what you do. And we were lucky to have like a pretty diverse group of investors as well, and so I think we're doing okay, and excited to do more of that.
Is there anything that you want listeners to know about Aperture? And if they want to learn more, where can they find you?
Yeah, so they can find us on our website, aperturetx.com. They can find me and our company on LinkedIn. And, we're based here in the Bay Area, and, we travel quite a bit. And so you can definitely see us at like local events or big conferences. What I want them to know about Aperture, if you're technical, I really want to excite people about the genetics has been like super old, type of science, but it's getting sort of a, renewed interest. And I think, there are really good things coming out of, people sharing their genetic data and sort of, other people in biotechs and pharma actually having a different type of looks using, machine learning AI and trying to figure out how we can design therapeutics. And so I'm very optimistic about where the field is going and sort of the new therapies that we will see like within the next, 5 years. And generally about Aperture, just follow us.
Yeah, totally.
Well, thanks so much, Martin, for coming on the podcast.
Thanks
we'll
for
do
having me.
Another one after the Aperture IPO. And,
all right. A couple years from now.
Sounds good. All right. Well, thanks so much, man. And keep up the good work.
Right. Thanks.
Thanks everyone for
guys.
listening. Bye.
Bye.